Hello, Charles William Amir Mansour here, scientist and global business development manager for Agilent Technologies. In this short video, I'd like to walk you through one of many target applications driven by our quantitative imaging and microscopy solutions. Accurate quantification of cell population growth, or proliferation, is an essential readout for many types of cell-based research, notably cancer-associated studies. Most current cell proliferation assays rely on indirect biochemical metrics that are limited by artifacts or image-based endpoint measures. Also, most fluorescent dyes that are typically used for cell counting have problems with inducing phototoxicity. In this example, wells are kinetically monitored every 2 hours using high-contrast bright field. This approach results in refraction of light by each individual cell to produce a bright spot, enabling accurate cell counting without the need for nuclear staining. Images are then processed to maximize contrast and analyzed as cell objects using intensity and size gating. The subsequent cell counts are then plotted against experimental conditions, for example, the concentration of an anti-proliferative agent such as doxorubicin. Another means to assess cell growth is to measure the percentage of surface coverage occupied by cells in the field of view, or confluence. Using high-contrast bright field, unlabeled cells can be distinguished from uncovered surfaces. This method is particularly effective for cell lines that do not grow as individual cells, such as MCF7 breast cancer cells, or with cells in suspension, such as K562 bone marrow lymphoblast leukemia cells. Live cell cultures can be monitored kinetically over several days, with confluence being automatically calculated by Gen 5 software. The subsequent cell counts can be plotted against experimental conditions. Thank you for taking the time to learn more about our application workflow. Feel free to explore other application areas through this video series.